We will continue our discussion with the molecules of living systems. Now, in the first lecture, we introduced the concept of these molecules, which are collectively called biomolecules. And we saw that these are basically carbon compounds. And we mainly discussed the structure of the carbon compounds in the context of stereochemistry. And also we discussed the various stereoisomers possible among the carbon compounds and their biological importance. So, stereospecificity, that is the ability to distinguish between stereoisomers. They are exhibited by many biomolecules such as enzymes and various other proteins, and is a very important characteristic feature on which the molecular logic of life is developed. Say food. We know that food is largely made of one particular mirror image form of the monomer. Say L amino acid makes the proteins, the polypeptides. Now, if food was made of molecules with the unnatural mirror image form, we would starve, because the enzymes in our bodies that digest the food, they are themselves chiral and have evolved in such a way that they preferentially recognize only the natural mirror image form of their substrates. This is a hypothetical diagram that shows the interaction of the different enantiomers of particular amino acid to the binding site. You can see that the binding site is asymmetric itself. Now, this enantiomer can fit onto the binding site, okay, but this one cannot. So, this enzyme can act on this isomer, but not this one. Now, the odor or the smell of many molecules, they depend not on their chemistry, there is a chemical composition, but on their overall molecular shape. Say these two molecules, hexachloroethane and cyclooctane. They are chemically totally unrelated. The chemistry is totally different, but their overall shape is similar, that is, they are both spherical. Consequently, each molecule, they can bind to the spherically shaped olfactory receptors present on the nerve endings in the nasal passage, resulting in similar odors. Both these compounds having totally different chemistry, but have camphor-like odor.
Sir, can two stereoisomers having the same chemistry have different odors?
Yes, of course. Let's see one example. Say these two molecules, they are enantiomers, S-carvone and R-carvone. Okay? Since they are enantiomers, chemically they are same. They have the same functional groups connecting the same way, but their stereochemistry is different, and their olfactory receptors are also chiral. So, S-carvone will be recognized by its own olfactory receptor and will give a smell like a caraway seeds. On the other hand, R-carvone, when recognized by its own chiral olfactory receptor, will give spearmint-like flavor. So, this is a contrasting example than the previous one, where two molecules having the same chemistry exhibits totally different smell. Now, stereoisomers are also distinguishable by taste. Let's take this molecular pair. Say here there are two stereocenters, and here also there are two stereocenters. Now, the configuration of this stereocenter is the same in these two molecules, but the other one has different configuration. This has a mirror image stereochemistry with respect to the other. Now, they are consequently recognized by different taste receptors, which are also chiral. That's why this has a sweet taste. On the other hand, this exhibits a bitter taste, though chemically they are totally similar. Okay, this sweet one is actually an artificial sweetener, aspartame, sold as NutraSweet. The same applies to the action of drugs in humans. For example, these two drug molecules, they are enantiomeric with respect to each other. One is called Darvon and another mnemonically is called Novrad. Okay, where Darvon acts as a painkiller, Novrad acts as an anti-cough agent. Now, the versatility of biomolecules, particularly the functional versatility, largely dependent on the presence of a variety of groups of atoms called functional groups that are attached to the carbon skeletons of the biomolecules and impart special chemical properties to the molecule. Okay. So, this actually justifies that the bonding versatility of carbon is a major factor in selection of carbon compounds, that is organic compounds, as a molecular repertoire of cells during the origin and evolution of living systems. No other chemical element other than carbon has been found to form molecules of such versatility in terms of size, shape, or a variety of functional groups, and hence the variety of chemical properties. Okay. And let us look at the different commonly found functional groups present on the biomolecules. These are the hydrocarbon functional groups, methyl, ethyl, phenyl. These are the oxygen containing functional groups, hydroxyl, carbonyl aldehyde, carbonyl ketone, and the carboxylic acid group. These are nitrogen containing functional groups, amino, amido, guanidino, imidazole, sulfur containing like sulfhydryl, disulfide, and the phosphorus containing the phosphoryl group.
So can different functional groups combine to form new functional group?
Exactly. Like here, this is a ester functional group. It has been formed by the combination of carboxylic acid and alcohol. Thioester, carboxylic acid and sulfhydryl group. Then ether, 2 alcohols. Anhydride, 2 carboxylic acid groups. Mixed anhydride, combination of carboxylic acid and phosphoric acid. Phosphoanhydride, combination of 2 phosphoric acid. So new functional groups can be generated by combining different other functional groups. Now, some biomolecules are special in the sense that they are polyfunctional. They contain more than one functional groups, each with its own chemical characteristic and reactivity. Consequently, the chemical personality of such polyfunctional molecules are determined by the chemistry of the functional groups in combination and of course their stereochemistry. Say histidine, amino acid found in the proteins. You see carboxyl group, amino group, imidazole group, 3 groups. Epinephrine, a hormone, methyl group, secondary amino group, hydroxyl group, phenyl group. Acetyl coenzyme, carrier of acetyl groups in many metabolic reactions. This is the acetyl group that they carry, this C3CO group. See so many functional groups, thioester, amido, hydroxyl, methyl, phosphoanhydride, imidazole, amino, phosphoryl. The mechanisms of biochemical reactions still could be deciphered from the nature of the functional groups of the reactants. The functional groups, what they do is they cause change in the electronic distribution and the geometry of the neighboring atoms, and consequently leads to the chemical reactivity of the entire molecule. Now, although there are a large number of different biochemical reactions happening within the cell, fortunately they could be classified into only a handful of general categories. Now, it's important to know that almost all the cellular reactions, they are enzyme catalyzed. Okay, the significance is that this allows the reactions to happen under cellular conditions, the cellular pH, temperature, ionic strength, etc., as well as with very high efficiency, very high precision, and of course a good control. Now, the different types of biochemical reactions within the cell could be classified into about 6 general categories. For example, oxidation reductions or redox reactions, then cleavage and formation of carbon-carbon bonds, intramolecular rearrangements, group transfers, condensation reactions in which monomeric subunits are joined to form biopolymers, and the reverse, that is the depolymerization of the biopolymers. Now, reactions within each general category usually occur by similar or comparable mechanisms. Let's start with the redox reactions, which are always involve electron transfer. Let's take an example, a type of redox reaction where a molecule loses a certain number of electrons accompanied with the same number of protons. So, effectively a certain number of hydrogen atoms are removed and the molecule is oxidized. The classical example of oxidation of lactate to pyruvate. See, in this particular group, from this group, 2 electrons and 2 protons are extracted. That means effectively 2 hydrogen atoms are extracted. So this alcohol group is oxidized to a ketone group. Okay. This electron acceptor in this case is a cofactor, nicotinamide adenine dinucleotide, and the cofactor itself is reduced. Now this reaction is perfectly reversible. It's catalyzed by lactate dehydrogenase, and this enzyme can also catalyze the conversion of pyruvate to lactate. And in this case, pyruvate has to be reduced back, and the electron will be added to the carbonyl group from the reduced cofactor. Now there are many biological oxidations where the carbon atom gets covalently bonded to an oxygen atom. The enzymes catalyzing these type of reactions are called oxidases. And sometimes in this type of reactions, the molecular oxygen is involved, and those enzymes catalyzing those reactions are called oxygenases. Now, importantly, we have to remember that every oxidation is always accompanied by a reduction, in which an electron acceptor acquires the electrons removed by oxidation, because electrons cannot remain free. Now, the biological significance of oxidation reaction is that living cells, they obtain the energy needed for the cellular processes from oxidizing metabolic fuels such as carbohydrate or fat. Most of the cellular catabolic, that is energy yielding pathways, are sequences of oxidative reactions that transfer electrons from these fuel molecules through a series of electron carriers and finally to molecular oxygen. The reason is that oxygen has very high affinity towards electrons, and consequently the overall electron transfer process is highly exergonic, and the energy obtained in this way drives the synthesis of ATP, which is the ultimate purpose of catabolism. Now, cleavage and formation of carbon-carbon bonds. First, the cleavage or breakdown, they can be of 2 types, homolytic and heterolytic. The homolytic cleavage, the electron pair that is being shared between 2 carbon are evenly cleaved, so 2 carbon radicals are generated. The examples of these type of free radical mediated reactions in biology are very rare. Much more common is the heterolytic cleavage, where one of the carbon after the cleavage retains the pair of electrons. Consequently, it is in excess in 1 electron and will contain 1 extra negative charge and will be called carbanion. And the corresponding carbon atom which has lost its electron is deficient in 1 electron and will carry 1 positive charge and will be called carbocation. Now, these carbanion and carbocations, they are themselves quite unstable and has to be stabilized by certain functional groups attached to them. For example, carbanion could be stabilized by electron withdrawing groups attached to it. On the other hand, carbocation could be stabilized by electron pushing groups attached to it. Now, this cleavage and formation of carbon-carbon bonds are usually mediated through nucleophilic substitution reactions. Okay? Nucleophiles are functional groups rich in electrons and capable of donating them, and electrophiles are electron deficient functional groups that seek electrons and will accept the electrons donated by the nucleophiles. Like here in this case, a nucleophilic substitution reaction, electron rich nucleophile Z attacks an electron poor center or electrophile, this carbon atom here, and displaces a poorer nucleophile W, which is called the leaving group. Okay. Most of the carbon-carbon bond formation and cleavage are mediated through this type of mechanism. Here is a list of nucleophiles and electrophiles that occur in the cellular conditions, like the O- ion, S- ion, C- ion, uncharged amine group of nitrogen, imidazole nitrogen, hydroxide ion, nucleophiles. Electrophiles will be carbonyl carbon, imine carbon, where the imine group has been protonated, then the phosphorus atom of a phosphate group and the proton. Okay. Interestingly, here you see, here carbon is acting as a nucleophile as well as here carbon is acting as electrophiles. That means whether carbon will act as a nucleophile or an electrophile will depend on the functional groups that are attached to the carbon in question. Some examples of this type of reactions: aldol condensation. You see this carbon-carbon bond is formed here. Example is the aldolase reaction. There is a reverse aldolase reaction actually happens in glycolysis, where 2,3 carbon units are joined to form a 6 carbon unit. Now, this carbon ion is stabilized by the presence of this carbonyl group with which this electron is delocalized. We will see this stabilization theme, this role of this carbonyl group is repeated in many biochemical reactions. Claisen ester condensation involved in the synthesis of citrate in the citric acid cycle. A carbon-carbon bond is formed here. Again, the carbon ion is stabilized by the same strategy. Now, an example of carbon-carbon bond cleavage. Here, this carbon-carbon bond is cleaved and this carboxylate group is liberated as carbon dioxide. Now, in this reaction again, the developing carbon ion is stabilized by the same strategy. Intramolecular rearrangement reactions, which involves electron transfer within a molecule. It is actually redistribution of electrons within the molecule itself, results in isomerization or transposition of double bonds and cis-trans arrangement of double bonds. We will check this isomerization reaction. Say glucose 6-phosphate is isomerized to fructose 6-phosphate and the enzyme is phosphohexose isomerase. See here this carbon 1 and 2. In glucose, this carbon 1 was a carbonyl group carbon belonging to an aldehyde group, and carbon 2 is an alcohol. After the isomerization and this rearrangement, carbon 1 has become alcohol and carbon 2 is now containing the carbonyl group. So, these two groups, the carbonyl group and the hydroxyl group, they have just rearranged between the two successive carbon atoms. It is an isomerization reaction or rearrangement reaction. Group transfer reactions. They are very important in activating otherwise inactive metabolic intermediate. The purpose is attaching a good living group to a metabolic intermediate to activate the intermediate for the following reaction. Okay. Now, for this purpose, inorganic orthophosphate and pyrophosphate are frequently used because they are much better and one of the most common living groups in nucleophilic substitution reactions involved in metabolic pathways. Now, nucleophilic substitution is made favorable by attachment of, say, a phosphoryl group to an otherwise poor living group such as OH. Okay. Now, in the phosphate group itself, since the oxygen is much more electronegative than phosphorus, the sharing of electrons is unequal. The central phosphorus bears a partial positive charge and therefore can act as an electrophile. Example of a group transfer reaction. See glucose with its hydroxyl group. This is the hydroxyl group attached to the 6th carbon. Itself is not that much active because this OH group is a very poor living group. Consequently, glucose in this form cannot participate directly in the nucleophilic substitution. So, what is done is a phosphate group is transferred to this. So, this OH group is a nucleophile and this phosphate with its partial positive charge is an electrophile. So, this will attack this and displace the rest of the molecule as ADP, and then the phosphoryl group will be attached to this OH group. Now, it has become glucose 6-phosphate and this is a much better living group and glucose 6-phosphate can immediately participate in the next metabolic reaction. These type of enzymes like hexokinase in this case, which catalyze the transfer of phosphate group from one molecule to another, is called kinase. They belong to a large family called kinases. Now, thiols and thioesters are also similarly used in many other metabolic reactions, for example, lipid metabolism. Now, the condensation reactions which lead to the formation of biopolymers. In these type of reactions, the monomeric subunits that constitute the biopolymers such as proteins, nucleic acid, polysaccharides, they are joined by nucleophilic displacement reactions that replace a good living group. Now, let us first consider a case, a hypothetical situation, joining of two amino acid molecules, say two glycine molecules, to form a dipeptide such as diglycine. Now, simply what can happen is that nitrogen can attack this carbon center nucleophilically and displace the OH group and form this bond, this so-called otherwise theoretically possible bond with the removal of water. But this does not happen because this OH group is a poor leaving group. Consequently, this is a very necessary reaction, the formation of peptide bonds or linking to amino acids. So, this OH group has to be activated. The cell has to solve this problem, and that is done by attaching tRNA, transfer RNA, about 75 nucleotide long small RNA, to this OH group, okay, to form an activated amino acid. This forms an ester linkage to the alpha carboxyl group of the amino acid that activates the carboxyl group for condensation with the alpha amino group of the other amino acid. So, now the nitrogen as nucleophile will attack this carbonyl carbon and displace the tRNA efficiently to form the peptide bond. Okay. Similar strategies are employed in the biosynthesis of other biopolymers such as nucleic acids and polysaccharides. Now, the reverse reaction, hydrolysis of the biopolymers, that is depolymerization of the biopolymers to smaller subunits. These macromolecules can be broken down by hydrolysis reactions, in which water is the attacking nucleophile, displacing a monomeric subunit or a smaller polymer fragment. The enzymes that catalyze these type of reactions are called hydrolases, and they are very much essential, particularly in the digestive process. Other examples are regulation of the level of critical macromolecules such as proteins and messenger RNA, because the critical level of a functional biomolecule such as protein and messenger RNA not only will depend on its synthesis, but also on its degradation. This is a reverse reaction. This is a peptide bond that has to be cleaved in this polypeptide. So, water is attacking as a nucleophile on this carbonyl carbon and displacing this part of the polypeptide. So, eventually we have two peptide fragments where the alpha amino group has been generated in this fragment and the alpha carboxylic group has been generated in this fragment. Okay. So, in this part we have discussed so far the importance of stereoisomers in biological activity, and then we discussed the different types of functional groups present in the biomolecules which determine the chemical properties of the biomolecules, and finally we discussed the 6 major classes of biochemical reactions that happen within the cell.